In the module on protein structure and stability, we will begin our discussions on the thermodynamics of protein folding. This is an important aspect and it encompasses a lot of the interactions and the studies that we have seen earlier. So, if we look at the concepts that are going to be covered, they are going to be protein folding in general and the thermodynamics of protein folding. And this is going to have an important aspect in the conformational entropy that we will see as we go along. So, as we saw that the factors that drive protein folding are hydrophobic collapse that results in the formation of the hydrophobic core. We have the polar surface that interacts with the solvent. We have a minimum volume that is occupied so that it is a compact structure. Then we have the stabilization by the other covalent bond formation, that is the disulfide bonds. Now, the additional aspect is hydrogen bonds that can occur, as we saw in the secondary structural elements, between the C double bond O and the NH of the backbone. In addition, we can have these hydrogen bonds between the side chains, and another is with the polar solvents. So, considering that this is an important aspect, each of these are going to contribute thermodynamically to the aspects of the protein folding that we are going to see. And of course, we have the polar and the electrostatic interactions, usually between the ion pair interactions that we see between the acidic and the basic amino acid groups. Now, the native state is typically only 5 to 10 kilocalories per mole, more stable than the unfolded form, which we will be discussing. Now, when we look at the hydrophobic effect, what is it exactly that we mean? It means that this is a tendency of non-polar surfaces to interact with each other to sequester themselves away from the solvent. So, the hydrophobic collapse that we see of the non-polar side chain is essentially to build this core of the globular proteins. Why? Because the van der Waals interactions that we see are favorable, but nevertheless the hydrophobic collapse is unfavorable for water molecules because they have to arrange themselves around the non-polar water surface. But we have to remember that we are looking at amides here. Each peptide bond is an amide bond that is linked by a C double bond O and an NH. So, this can actually interact with the water. So, when we are looking at the hydrophobic collapse of a polypeptide chain, this leads to the increased entropy of water, in the sense that the water that had been collectively around the polypeptide chain in its unfolded form now has to rearrange itself to form the polar contacts with the molecules, or rather with the residues that are on the surface of the protein. And in addition to what happens to the core of the protein, there are these hydrophobic amino acid residues that tend to move away from the surface of the protein. So, what happens is there is a balance between the hydrogen bonding and the unfavorable steric and electrostatic interactions that follows the precise shape of the protein, and most of the water molecules in this case are eliminated from the interior of the protein. But nevertheless, there are some cases where we see what are called structured water molecules that are hydrogen bonded in such a fashion that they are more or less rigid in their mobility in terms of being a water molecule, or with respect to the ones that we can see on the surface that are more flexible in nature. So, when we look at the non-polar amino acid residues, for example, phenylalanine, leucine, isoleucine, valine, methra and tryptophan to a certain extent, they tend to cluster in the interior of the protein, and the other amino acids are preferably near the surface of the protein so that they can interact with the water and other polar moieties by hydrogen bonding formation. So, if we look at this non-polar solute now, we observe that there are hydrogen bonding network around this non-polar solute, and so what we mean by this particular possibility is that the tendency for the non-polar surface to interact with each other rather than water, that is going to lead to the burial of the non-polar side chains is what is termed as the collapse. So, the hydrophobic side chains that are there in this unfolded protein will tend to come together in a collapsed form to form the core of the protein from an extended coil that did exist. So, if we look at it from a hydrogen bonding standpoint, it is not actually very favorable. Why? Because the hydrogen bonds between the water molecules that were there, as we can see here, they have to be disrupted because of this non-polar molecule that is there. And what happens is the unfavorable situation results as the water molecules need to become highly ordered to bring about or to sequester themselves away. When the non-polar part sequesters itself away from the water, then the rest of the water molecules have to arrange in an ordered manner around it. So, this gives an entropic aspect to this. So, when we are looking at a non-polar solute, this is what we see. When we look at a polar solute, we know that the polar molecule or the polar moiety itself has hydrogen bonding capability with the water molecules. So, the dissolution of the non-polar solvent is water is unfavorable, and for polar solutes we have favorable hydrogen bonding. So, when we look at the other structure stabilizing interactions, we see that there are strong dipole-dipole interactions, which we would expect with the water molecules, and of course, again with the hydrogen bonding that we see in the C double bond ONH of the peptide linkages throughout the protein. So, whether we are looking at a beta sheet or the strands of the beta sheet connecting together, or in the alpha helix, we see this sort of interaction between them. The other salt bridges that are observed are between the acidic and the basic amino acid side chains. In addition to this, we see van der Waals interactions and hydrophobic interactions. All of these are structure stabilizing interactions. So, now if we look at the thermodynamics of the protein folding, we realize that when we have the unfolded form that is completely in a random state, then what is going to happen? There are going to be many such conformations possible. Now, each of these have the peptide bond that is linking the different amino acids together. So, they have a water network around it. But then when we have the hydrophobic parts come together in the collapse, this water is disrupted and we have the entropy that is going to be large here gradually decrease in terms of looking at from a protein standpoint. So, this protein molecule is now going to fold into a compact form and it is going to form a native structure. So, this native structure gives you different folding intermediates in between, a molten globule state that says there are some elements or some condensation, in the sense that some elements or some nucleation occurs where we have some beta strands form to form beta sheets, a part of a helix and so on and so forth, and then we have the compact native structure. So, initially when we have the native conformation, we have the polypeptide chain, we have a large number of possibilities that gradually then get to a minimum energy state to give us the native structure. So, when we look at the protein folding, it occurs by diverse pathways, and there are components, as I mentioned, of a nucleation and a condensation mechanism that actually describes the overall features of folding of most of the domains that we know about. So, if we are looking at the unfolded structure in this form, and as we know, we look at a protein folding funnel, we have the final folded structure. So, the unfolded structure and the folded state are in an energy form where we have the folded state at a minimum energy form that is going to be the compact structure. But there is much more to the protein folding aspects or the thermodynamics of the protein folding that will be covered in this course. So, there are two important components. So, here is our unfolded structure now, and we have the peptide hydrogen bonds and the peptide solvation that are interconnected. So, the hydrogen bonds that we saw between the water and the peptide NH and CO, because now this is the unfolded form that is on the right here, they have to be broken before the peptide H bonds themselves are formed. Okay, so the NH CO have to be broken. So, the peptide desolvation caused by the folding is an exchange reaction that is accompanied by H bonding, hydrogen bonding of the buried peptide NH-CO groups that results in a large energetic factor. So, this is important in the peptide solvation that we are talking about and the hydrogen bond formation that we are talking about. So, if we look at the overview of protein folding, we look at an amino acid sequence of a 56 residue peptide and we see the steps that are involved. So, the specific sequences, the amino acid sequences, they fold into local secondary structures. What do we mean by this? We have certain regions that are marked in red here and we see that this part has formed a coil or a turn like this. This part is a random or rather a helix, and then this part then gradually gets into strand formation to form a beta sheet. So, then what happens is there is the assembly of the local structures that is followed by the long range interactions to give specific types of motifs that we looked at. For example, we can have a beta alpha beta motif or an alpha beta alpha motif. So, that would be again a secondary structure elements coming together for say something like this, where we would have a beta sheet and we would also have an alpha helix. So, then what happens is while this is happening, the hydrophobic interactions constantly sequester the non-polar amino acid side chains in the protein core to give its final tertiary structure. So, while this is occurring where we are having the hydrogen bonds being formed, the water molecules being pushed out from the central core of the protein so that the hydrophobic interactions may take place, then that is going to give rise to the protein core. Now, we have computer programs that have been developed that can often predict the structures of proteins on the basis of this information. So, on the basis of the amino acid sequences, we might be able to say that given this sequence, because we know from Anfinsen's experiment to a certain extent that all the information that is required for the three-dimensional structure of the protein is present in this amino acid sequence. So, if we look at the thermodynamics of the folding, now we have our unfolded structure. We remember again that not all of some of these on the surface are still hydrophobic in nature, but it gradually gets to a nucleation and a condensation, giving us the folded structure. So, we have, if we want to look at now the free energy of folding, it would be the final form, that is the G folded minus the G unfolded. So, we look at the unfolded formation and we see that it is disordered, the entropy is high. On the other hand, if we look at the folded conformation, it is an ordered conformation. So, the entropy now is low. If we look at the formation, we know that the free energy delta G is given by delta H minus D delta S. So, if we look at the delta G of folding, we have to look at a delta H of folding and a T delta S of folding. So, the contributions are going to come from, so if we look at now what the delta S of folding is going to be in terms of what we see here, it is going to be the S of the folded minus the S of the unfolded, and the delta H of the folding is going to be the H of the folded minus the H of the unfolded. So, if we look at this description at equilibrium, we have this is what we are looking at. We have the native state and the unfolded state. This is the reverse of, so this is unfolding that we are looking at, and the equilibrium constant that we have here is the K value that is given by the concentration of the unfolded component divided by the concentration of the native component, which we will see when we look at protein denaturation as well. So, the difference between this gives us the delta G of the unfolded and the native states. Given that this is in equilibrium, we have an equilibrium constant associated with this, and we have the delta G that we can calculate, and from this we can actually get the delta G of the folding process. So, when we have a K unfolded, we have a positive delta G that indicates that the native state is more stable. Now, when we look at the spontaneity of the process and we say that the protein folding process is a spontaneous process, this means that this delta G value has to be negative. Now, if it is negative, and we know that from a protein standpoint, the flexibility that we observed in terms of delta S is taking it from a more unordered state, disordered state, to a more ordered state, giving us a delta S that is actually negative. So, what happens in this case, when we are looking at the minus T delta S form, it is actually a positive quantity. So, when we look at the bonding, the enthalpic contributions, we will say that the enthalpic contributions for the different bonds that are possible to form, they give us our contributions to the overall protein folding. So, what do we mean by this? So, again we have the unfolded protein, we have a delta H that is small and negative, we have a delta S that is large and positive. When we now look at the folded protein, we have the delta H that is large and negative and a delta S that is small and positive. Now, the folded part, we realize that the delta S is going to be small and positive, and the delta S that is going to be large and positive, because we have to remember that we are disrupting the water molecules as well. So, there is a conformational entropic contribution there. So, the compensation in entropy and enthalpy for the protein, there is a contribution of entry of the water molecules that is released upon the folding. So, the delta S of the water that we actually see of the surroundings is large and positive. So, all of this together is going to contribute to the overall delta G that we observe. So, if we look at the change in the delta G, or the change in the free energy, it is going to be negative for the spontaneous folding. So, for the spontaneous folding, what we are going to observe is we have the delta S of the system, which is our protein, that is going to be negative. Why? Because we had the unordered state, the disordered state that folded into an ordered manner, giving us a delta S that is negative. So, the delta H of the protein is also negative, because we have all the enthalpic contributions from all the possible interactions, the hydrogen bonds, the electrostatic interactions, the van der Waals interactions, they all contribute to the enthalpy. So, the protein folding that we have is driven by enthalpy, but the delta S of the surroundings is much greater than 0. So, we have the delta G that is negative, and the hydrophobic effect that plays a very important role in the protein folding process. So, if we look at the contributions to the delta G of the folding, it is the delta H of the folding minus the T delta S of the folding, and we found out that if we want to look at the delta H of the folding, we have to look at H folded minus H unfolded. When we look at the delta S of folding, we have to look at the S of the folded minus the S of the unfolded, because when we are looking at folding, our final form is the folded form. So, if we look at the contributions now here, when we are looking at a delta S of folding, we see that this contribution is negative, meaning that we have a negative contribution to the delta S, because we are going from a disordered to an ordered form. So, when we look at the expression in the minus T delta S, this becomes a positive quantity in the minus T delta S contribution. However, when we are looking at the delta H of folding, that is the enthalpic contributions to protein folding, there are all contributions that are going to add up. What are these contributions? The contributions correspond to the hydrogen bonding that are possible between the polar entities, the hydrophobic interactions that are possible between the nonpolar entities that are going to form the hydrophobic core of the protein. We have the ionic interactions that are possible. We also have the disulfide bond formations that are possible between the cysteine residues. And what is going to happen here is all these are going to contribute to the overall enthalpy of the folding that we see. And in effect, when we look at the delta G folding and we look at the delta H of the folding minus the T delta S of the folding, we see that the enthalpic contributions that we see here give us this small negative factor for the delta G folding that makes the delta G folding process be negative, which means that this is a spontaneous process. So, the contributions that we see are extremely important. So, what are these contributions now? If we can summarize what we have learned, we have the hydrophobic effect. This hydrophobic effect is the release of water from the structured solvation layer as the protein folds that increases the net entropy. So, we have to be very careful here when we are considering as to why are we considering an increase in the net entropy, because we are looking at the water molecules that are being moved away from the solvation of the peptide, and now this is getting folded because we have the hydrophobic interactions occurring between the nonpolar residues that would tend to remain away from the water, tend to remain away from the solvent. So, this is what the hydrophobic effect is. So, it is the release of the water from the structured solvation layer as the protein folds that results in an increase in the net entropy. But from a protein standpoint, when we are looking at the disordered polypeptide chain, when that is getting folded into a more ordered compact structure, that is resulting in a decrease in entropy for the system, that is our protein chain. When we look at the hydrogen bonds, we looked at the interaction of the NH and the CO, which we know, of the peptide bonds leading to the local structures such as alpha helices and beta sheets. So, in the overview of the protein folding that we saw, there are the formation of the alpha helices, formation of the beta strands that are forming the beta sheets that result in the local structures, and then we have the formation of the domains that are then going to lead to what we call the molten globule state. This molten globule state is going to gradually get to an energy minimized form, where we have the native state being the most stable state that we know as the folded state of the protein. In the London dispersion forces, we have medium range weak attraction between all atoms, and this also contributes greatly to the stability in the interior of the protein. We have to remember that this is the dipole-dipole interactions, so there is an instantaneous dipole that occurs in all atoms, which brings about this van der Waals types of interactions. We have the electrostatic interactions. Now, the electrostatic interactions are these long range strong interactions between permanently charged groups. So, which are these permanently charged groups? These are the ones that have the positive charge or the negative charge. So, we have the basic amino acid side chains, we have the acidic amino acid side chains, and the interactions between these are strong interactions, and these ion pairs that are formed, the salt bridges, especially buried in the hydrophobic environment, strongly stabilize the protein. So, when we have a specific type of amino acid residue that belongs to the charged polar type, that is the basic or the acidic type, they would prefer to be on the surface of the protein. But if it so happens that they are in the hydrophobic environment of the protein, in the hydrophobic core, there is usually a salt bridge occurring where we have an interaction between the positive amino acid and the negative amino acid in the formation of the salt bridge. Again, we can look at the water molecules that are present within this, where sometimes we have bridged water molecules that have a specific structured characteristic to them because they are involved in hydrogen bonding and they are caged within the protein structure. But those on the surface are more flexible because they can exchange themselves with the bulk water. So, the different references that we have followed here are the specific books and specific items also, and in addition we will revisit protein denaturation to see what the structural aspects occur, when the protein loses its native structure. Thank you.